Hello and welcome to Nicholas Genetics Lessons, and today I have prepared two multiple choice questions for you, and as usual I recommend you to stop video here, try to solve each problem on your own first, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. And here is the first question. The ability of some tRNAs to bind to more than one codon is called, and here is the four answers to choose from. Answer A, slope, B, ionizing, C, wobble, and D, fluctuation. And the correct answer is C. So this is, you just have to remember that third position in many codons. So as you see here, we have two codons that code for one protein, two here and four here. But the third position in many codons is not as important as the first two positions. So we call this third position wobble position because the same tRNA can bind and make non-classical pairing where we have, for example, uracil always pairs with adenine and cytosine with guanine. So our organism and many other animals and plants don't require 64 different tRNAs, or actually 61 because 3 would code for the stop codon, and that means that at this place special protein binds. So 61 tRNAs would need if not for the wobble position of the third nucleotide. But in reality we have much less tRNAs, and in the middle of the 1960s Francis Crick proposed the theory that third position is not as important, and he calls this wobble position. Now let's move to the second question. A synthetic messenger RNA is composed of 3 parts uracil and 1 part of cytosine. This messenger RNA would code for a protein that contained approximately what proportion of the proline. So it is abbreviated as PRO, and here's the answers. And once again, in order to solve this problem, we would need a codon table. So here is the codon table. So we are looking for proline, and here is the proline here. It is coded by 4 codons or 4 three-nucleotides. Let's write down these sequences. So the first one would be C, C, U. The second one would be C, C, C. The third one would be C, C, A. And the fourth one would be C, C, and G. So we don't need this table anymore, so we can now work with this information that we got. According to our problem, we have only 2 kinds of bases. This is going to be 3 parts of uracil and 1 part of cytosine. So let's now return back to these 4 codons. And actually, because we have only uracil and cytosine, we wouldn't be able to make with these bases this three-nucleotide, because here we have guanine. So we can cross out this variant, and here we have adenine, and we also don't have this base, so we can cross out this variant also. So only these 2 three-nucleotides can make proline. In our next step, we have to find how many different combinations we can make with 2 bases, with uracil and cytosine, when we make three-nucleotides. And the first variant would be when we would have 3 uracils, so uracil, uracil, uracil. The other variant would be when we have 2 uracils and 1 cytosine. So this is can be uracil, uracil, cytosine, uracil, cytosine, and uracil, and cytosine, uracil, uracil. And next combination could be cytosine, 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 And the rest combinations can be when we have two cytosines and one uracil. So cytosine, cytosine, uracil, and cytosine, uracil, cytosine, and uracil, cytosine, cytosine. As you can see, with two types of bases, we can make 8 different combinations of trinucleotides. But not all these combinations would code for the proline. Only these two combinations, CCU, and this is first combination, and the other one would be CCC, and this is the second combination. If we would have equal number of uracils and cytosines, these 8 combinations that they can make, and the frequency of the proline would be 2 out of 8, or 1 out of 4, or 25%. But because uracil present as 75%, or 3 parts, and cytosine present as 25%, or 1 part out of 4, we also have to make one more step in our calculations. So imagine that we have first position, second position, and third position in our trinucleotide. If we would have equal number of uracil and cytosine, if I would ask you what is the probability that the first position would be taken by uracil, your answer would be 1/2. And if I would ask, for example, what is the probability that second position would be taken by cytosine, once again, your answer would be 1/2. And for example, third position would have equal chances to be whether uracil or cytosine. So any of these bases have 1/2 chances to take this position. But because we have different frequency of these two bases, if I would ask you what is the probability that we would have uracil here, your answer would be 3/4. This would be probability, and 1/4 probability would be that this is going to be cytosine. So this is going to be probability of the uracil. And if I would ask you what is the probability in the second position to be cytosine, you would say this is going to be 1/4 for the cytosine. And if I would ask you what is the probability for the third position to be uracil, once again, you would say that probability would be 3 out of 4. So we have, for example, uracils and cytosines, and uracils are present in much higher frequencies than cytosines. Per 1 part of cytosines, we have 3 parts of uracils. And of course, if we would take randomly blindfold any of these bases and would put randomly here, this is going to be probabilities that would get this combination, for example. So in order to find probability for these two proteins to be made with this frequency, initial frequency of our bases, we have to calculate probability for each position. So for the first variant, this is going to be 1/4. We have to multiply by 1/4 and by 1/4. I don't have much space left, so the answer as a decimal number would be 0.016. And for the second variant, we also can calculate probability. This is going to be 1/4 for the first position to be cytosine, and 1/4 for the second position to be cytosine, and 3/4 for the third position to be uracil. And the probability here would be 0.047. Now we have to add these two probabilities, and our answer would be 0.063. Or if we need an answer in percentage form, we have to multiply this number by 100, and our answer would be 6.3%. And let's now find our answer here. In order to do so, let's convert fractions into the decimal number. So here we would have for the 27/64, the fraction would be 0.422. For the answer B, 1/64, the answer would be 0.016. And for the answer C, 4/64, our answer would be 0.0625. And if we divide 3 by 64, we are going to get 0.047. And as you see, in my calculations, I used rounded numbers. And as you see, this number is very close to this one. So the correct answer would be answer C. And once again, if we would have only two bases, one would be 75% frequency of this base, another would be 25% frequency, and if we would make a random sequence of the message RNA, we can get 8 different combinations. Only 2 of these combinations would code for the proline, as you see, and frequency of each combinations we have found here, and this is going to be frequency of proline coded on this randomly synthesized message RNA. And of course, if you calculate frequency of occurrence of each three-nucleotide, and when you combine all these 8 variants, you are going to get 100%. And just if you're interested, for example, the frequency of this three-nucleotide would be about 42%, 3/4 probability for the uracil to take the first place, 3/4 probability for the uracil to take second place, and 3/4 probability for the third position to be taken also by uracil. So this would be most frequently occurring three-nucleotide in such message RNA, and we can find that almost 42% in such message RNA would be this combination. And the rest combinations each would be occurring in about 16%, and as you see, these 3 combinations would occur 4.7% each, and this one in about 1.6%. So if we combine all these frequencies, we are going to get 100%. But only these 2 combinations would code for protein proline. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.